Okay, so this chapter is about essentials of metabolism. You've learned a lot of the information in this already because it's got cellular respiration. It has enzymes. I mean, all these things that you should definitely already know. So we're not going to go over all of those. I'm not going to test you on them. Okay, two basic concepts just to remind you, right? Anabolism and catabolism make up metabolism. So catabolism is when we take larger molecules and we break them down into smaller molecules and energy is released. And anabolism is when we take smaller molecules and put them together to make larger molecules, and that requires energy. Okay, now microorganisms are super diverse. Okay, so when we talk about them, we're going to consider a bunch of different things. They're way more diverse than humans are. So they have energy they can derive energy from different places, and they can derive carbon from different places. Okay, so organic molecules such as glucose, fatty acids, and acetates are places where they can get carbon. They can also get energy from hydrogen atoms, hydrogen sulfide, and then iron. They can also function kind of like plants and do photosynthesis, so they can use light to make energy. Humans can't do this, okay? So glucose, fatty acids, acetate. So acetate's kind of like plastic. Hydrogen sulfide, iron, and light. Okay, now the two terms that we use to describe energy sources are chemotroph and phototroph. So a chemotroph depend on reduction, and oxidation reactions of inorganic or organic chemicals for energy. And then a phototroph, they're going to convert solar energy to chemical energy. Okay, now carbon sources for bacteria, so they could get from organic compounds like us. Okay, they could also get from methane, carbonate, or carbon dioxide. So way more diverse. Okay, so autotrophs. So troph has got to do with feeding, right? So if an autotroph is a self-feeder, so that means they can convert carbon dioxide into things like glucose. So they don't usually cause disease. And then heterotrophs, so hetero is other. They can feed on others, so they get all their carbon from organic matter. So it's usually from dead stuff. Okay, so we're an example of a heterotroph, whereas a plant would be an example of an autotroph. Okay, so if we look at this picture, this is from the book. So all microorganisms are broken down into autotrophs or heterotrophs. Autotrophs are self-feeders, so they make their own food by reducing carbon dioxide, and then heterotrophs use organic molecules that are already out there. So they can use light for energy, or they can use inorganic compounds for energy. So here's some examples. So green, purple, sulfur, cyanobacteria, algae are all examples of microorganisms that can do that. I want you to be familiar with the basic vocabulary. I don't expect you to memorize like what goes where. Okay, chemoautotrophs are going to make energy using other chemicals. Okay, so iron, sulfur, hydrogen, nitrifying bacteria. Okay, now photoheterotrophs are purple non-sulfur bacteria and green non-sulfur bacteria. And then chemoheterotrophs are like pretty much everything. This is the big one. Okay, so we're chemoheterotrophs. So what metabolism is found in most chemoheterotrophs? It's cellular respiration. Okay, that's where how we make our ATP is with cellular respiration. We're chemoheterotrophs. Okay, all of these other things are really cool. We just don't spend a lot of time talking about it in this class. Okay, so now microbes obtain their energy from food, so they use enzymes, and you guys know what enzymes are, right? They're proteins. They reduce the energy of activation. They can be reused over and over again. They depend on the active site being the right shape in order to work. Temperature, pH can affect them. Okay, now there's two categories of microbial enzymes. So there's endoenzymes, which these are made in the cell. They work inside the cell. So if you look at this little picture, they're in the cell, they're made in the cell, and it's just kind of the normal metabolic pathways that bacteria are going to do. Now, exoenzymes are also called permeases, and they're produced in the cell, but they cross the cell membrane and the cell wall, and they work outside the cell, or they work in that little space in between the cell wall and the cell membrane. Sometimes they call them permeases, so they permeate, they go through. Okay, so why exoenzymes? So I have a little video that you can go to. I'll put a link for it, and it just talks about exoenzymes. Okay, so pretty much what happens is that exoenzymes can break down big molecules or chemicals that might harm the cell. Okay, so they break down big molecules that the bacteria can actually use once they get inside the cell. So they're too big to get through the cell, so that the bacterium excretes the enzyme outside the cell. The big molecules get broken down into little molecules, and then the little molecules can be absorbed. Okay, sometimes exoenzymes break down things like antibiotics that might damage the cell. Okay, now they can also be used to help a bacterial pathogen avoid host defenses. Okay, so we'll talk about that. They are considered virulence factors because they make bacteria more pathogenic. Okay, we will use some of them to identify bacteria. So different groups of bacteria have different exoenzymes. So you can use the properties of the exoenzymes to identify the organisms. Okay, so here's examples. We already kind of went through this on the last chapter. So carbohydrate, sucrase, lipase. This picture shows gelatinase. Okay, so gelatin is a protein that it actually comes from horse hooves. Okay, so it's like jello, right? But there are some bacteria that make gelatinase, and it breaks down the jello. Okay, so it essentially just breaks it apart. Okay, now here's some important exoenzyme examples, especially related to how virulent pathogens are. Okay, so one is streptokinase. So streptokinase comes from Streptococcus pyogenes, which this causes strep throat. Okay, and what it does is it actually digest blood clots. Okay, so Streptococcus pyogenes causes strep throat, and it can cause skin infections. Okay, streptokinase is an enzyme, it breaks down blood clots. So if you think about it, I'm going to try to draw a picture up here. Okay, so say this is your skin, and you step on a, cut your finger with a piece of glass, and bacteria get in it. Okay, what your body normally does, okay, is it forms a blood clot around that to keep any of the bacteria that are in here from spreading throughout the body. So what Streptococcus pyogenes can do is it can produce streptokinase, which will break down the blood clot, and then the bacteria can circulate throughout the body. Okay, now streptokinase has also been used as medicine because it breaks down blood clots. Okay, so if someone has a blood clot or they think they're having a stroke, okay, they can administer streptokinase to break down the clot. Okay, lipase is produced by Staph aureus. Okay, so Staph aureus causes skin boils. Okay, and this organism it invades like where the hair goes in the skin. So it likes all of the oily parts. So it can break down the oils, and it can cause a boil. Okay, so it's breaking down the fat. Elastase and collagenase are from Pseudomonas. Okay, so Pseudomonas it's green, it smells It has a very distinct smell. I'll let you smell it in lab. But elastin and collagen are in the connective tissue, and if you have a Pseudomonas infection, they can get broken down. Okay, so people who are burn patients or people that have cystic fibrosis often have Pseudomonas infections, so they can start to have connective tissue breakdown. Okay, and I have these other two on here, but I just want to focus on these three. Okay, now this is going to talk about basic cellular respiration. Okay, so the first thing I want to remind you of is when we start breaking down glucose, the first step typically is glycolysis. Okay, in glycolysis, we go from glucose, we end up we make some ATP, okay, we make some NADH, and we make 2-pyruvate. Okay, glycolysis is anaerobic. Okay, it doesn't require any oxygen. Okay, so we end up, it's very low energy yield. So the NADH molecules are what's actually carrying the electrons around. Okay, but the pyruvate is going to be important. Okay, and the idea that this is anaerobic is important. Okay, so here's all the steps of glycolysis, and we're not going over those again. Okay, now what I want you to realize is that bacteria have some alternatives to glycolysis. Okay, so they have this one thing, it's called pentose phosphate pathway, or PPP. Okay, so what happens, instead of taking in glucose, which is a 6-carbon sugar, they can start with 5-carbon sugars. Okay, so which is a pentose, right? A 5-carbon. So it can take 5 carbons, okay, and it can start the whole pathway. So it can make ribose, it can make some amino acids, it can make NADPH, which you can use to carry energy around. Okay, there's another alternative, which is called the Entner-Duodorf pathway, and it's in Pseudomonas, and it's just another pathway. So they don't have to start with the 6-carbon sugar. That's the gist of it. That's what I want you to get. Okay, now fermentation is what happens if there's no oxygen available. Okay, so remember that fermentation is anaerobic. Okay, so what happens is it's just going to keep circulating the NADH so that you can keep using it for glycolysis. Okay, now pyruvate is going to be the first part of fermentation, and what we're going to end up with is we're going to end up with acids. We can end up with alcohols, and then we can end up, there's some other kinds of fermentation. Okay, so there are, here, let me, okay, if we look here, so we're coming in with glucose, and we have pyruvic acid. Okay, so then we can have homolytic acid fermentation. So this is stuff like lactobacillus. Okay, and we can take lactic acid, and that's how we get yogurt, that's how we get cheese. Okay, then we can have alcoholic fermentation. Okay, so that's how we get beer and wine and bread. Okay, then there's all these other kinds of fermentation. Okay, so there's mixed acid, propionic acid, butanediol, and then this butyric fermentation. Okay, now we can use these different kinds of fermentations to identify organisms. Okay, so we'll use these when we start to do biochemical unknown testing. You don't have to memorize them. I just want you to be familiar with the idea that there are all these different kinds of pathways. Okay, now here's the Krebs cycle. Okay, different organisms can go through different pathways. So there are nitrifying bacteria. Okay, so nitrifying bacteria are important in crop rotation. There's just a bunch of different things. So these chemoautotrophic bacteria are deriving their energy from chemicals. So they have totally different pathways, and you guys don't have to memorize them or anything. Just understand that they exist. Okay, now, what do bacteria do with the energy that they make? Okay, so they go through biosynthesis, so they make the chemicals that they need. They go through, they use ATP for membrane transport. Okay, they move. Okay, so they have motility. One another example is there are some that are bioluminescent. So there are some bacteria that will fluoresce. Okay, so they use their energy to light up. One example of that is the bacteria that's found in this little tentacle of the anglerfish. Okay, so that's actually bacteria that are bioluminescing that you see here. And another example is a squid, and I'll post a video, a little really cute little video about the squid too.